Welcome back to Chapter 5, Microbiologists. So let's talk about our enzymes a little bit more. We already discussed what an enzyme is. So you can tell me it's a protein. You can tell me it is not used up or changed in a reaction. You can tell me that it is able to decrease activation energy. But our cells need to be able to control enzymes, to be able to control what reactions are going on. To be able to, create the reactions we want happening, to inhibit reactions we don't want happening. And enzymes are also going to be really good target sites for a lot of drugs, because if you can inhibit an enzyme, you can stop a critical reaction from happening. You've just stopped that microbe from growing. So some factors that influence enzyme activity. One would be temperature. So as the temperature goes up, there's going to be more energy in the molecules. They're going to move a little bit faster. So the rate of reaction will go up until it hits its optimal temperature. The optimal temperature is where that reaction happens the best. But once it gets hotter than that, the enzyme starts to denature. Hope you remember that term, denature, from when we were in Chapter 2. That means it's going to lose its three-dimensional structure. And if you remember, structure is everything for a protein. Structure determines its function. So if that enzyme denatures, that reaction happens no more. Within our bodies, we as people have, and a lot of animals have, developed the ability to run a fever when we have an infection. Because as we run a fever, it starts to denature the enzymes that are allowing that bacteria to grow, or allowing whatever pathogen to grow, and so it helps inhibit the growth of that microbe. Another thing that influences enzyme activity is the pH. Most proteins have an optimal pH. Some proteins that are going to function in your stomach acid are going to need a pH of, like 2. If it's going to function on your skin, it's going to have a more neutral pH, slightly basic pH. So different proteins have ideal pH levels. But if it's too acidic or too alkaline, those enzymes can denature. Then next is substrate concentration. So for an enzyme to be doing anything, it has to be able to interact with a substrate. So if you don't have much substrate around, the enzyme's not doing much. As you keep adding substrate, then you'll have your enzyme acting more and more, until you hit a point of saturation. Saturation is when that enzyme is working the hardest it can. It just cannot go any faster. And if you start throwing in more substrate, you're just going to start making a line, just back up, traffic jam, waiting to get through. That little enzyme is working its hardest, and it can't do anything more. And then inhibitors. Adding an inhibitor will inhibit an enzyme. Taking away an inhibitor will allow that enzyme to start functioning again. So here's just a reminder of protein denaturation. If it's too hot, if it's too alkaline, if it's too basic, your nice functional protein, so maybe has an active site right here. If it denatures, you don't have that active site anymore, do you? It can't function as an enzyme when it's denatured. So let's look at some of these factors. Temperature. So here you can see as it's getting warmer, it's acting more and more. It's increasing its enzymatic activity, because more temperature means more energy. Until right here, right around 37 degrees or so is its optimal temperature. And it's going to be working about the fastest it can. I just lost my mouse. So it's going to work the fastest it can here. Beyond that, it stops. It just very quickly goes down. Do you remember what's happening at that point? That is when this enzyme is getting denatured. Too much heat is breaking all those bonds in the protein, so it's denaturing. Here, pH is affecting this enzyme's reaction. So here, right around pH of 5, is where this enzyme is most active. So would this be found somewhere acidic or somewhere basic? A pH of 5 is slightly acidic, moderately acidic. So it's going to be found in more acidic places. As it gets more to a more neutral or even into an alkaline pH, you can see it's probably denaturing as the reaction rate is going down. And as it gets too acidic, its reaction rate is going down. And here substrate concentration is affecting how this enzyme is reacting. So when you have no substrate, the enzyme's not doing anything. But as you start adding substrate, it starts working. It's working, and then bam, here are just flat lines. So the enzyme is not being denatured in this, right? There's nothing denaturing it. At this point, your enzyme is super busy. Just do a reaction. It's working as hard as it can. Throwing in more substrate isn't going to make it faster. It's the hardest it can be working. Let's go back to inhibitors. So to control how a microbe grows, all you have to do is control their enzymes. If you control their enzymes, it can't break down food sources. It can't get energy. It can't build up stuff it needs to function as a cell. The cell will die. There are two types of inhibitors, two categories of inhibitors: competitive inhibitors and non-competitive inhibitors. A competitive inhibitor is going to sit in that active site of the enzyme. So if that competitive inhibitor is sitting in the active site, the substrate will not be able to reach the active site. So it's just a straight up competition. It's going to fight for that space. So with the competitive inhibitor sitting in the active site, the substrate will not be able to be broken down into products. A non-competitive inhibitor is not going to sit in the active site. It is going to find somewhere else on that enzyme to bind, anywhere else. And wherever it binds is called the allosteric site. When the non-competitive inhibitor binds to the allosteric site, it causes the protein to just shift around, kind of make itself a little more comfortable. And as it shifts around and changes shape, that change in shape makes the enzyme non-functional. So even though a non-competitive inhibitor is binding somewhere else, it's making the enzyme respond a little differently. In both types of inhibitors, we are going to have no products formed. We have stopped the enzyme from functioning. So here is the protein, the enzyme, and its active site. Substrate binds right here. With a competitive inhibitor, it binds here to the active site. When it's bound to the active site, the substrate can't bind. So this reaction doesn't happen. Here, same enzyme again, but with a non-competitive inhibitor, it binds somewhere else. So it doesn't touch the active site. It touches this little spot on the side called an allosteric site. When it binds to the allosteric site, that enzyme says, ooh, that's a little uncomfortable, and tries to shift around. And when it does, it turns this active site. It's all kinds of altered. So your substrate can't bind here. So now you don't have substrate binding. You still don't have a protein. You still don't have the reaction being carried out. There's also a type of allosteric reaction called feedback inhibition. Feedback inhibition is a really important way that our cells are able to make sure that we don't create too much of something, that we're not wasting energy building something that we don't need to spend energy on. So as you take your substrate, it goes through a reaction, goes through all of these reactions, this whole metabolic pathway, and at the end you finally have your product. As your product builds up and you get a whole bunch of it, it can come back and interact with an enzyme at the start of the pathway and shut it down. So instead of just building up all of this product again and again and again when you don't need it, it can come back and say, okay, you made me, you did your task, now stop. It fills in this allosteric site, shuts down the reaction, so you're not building up extra. As the levels of that, or as it's not being built, the levels will start to fall, and that allosteric reaction will come back off, and the feedback inhibition will end, and you'll start the reaction happening. Then you'll start building up, and you'll have feedback inhibition going on again. So you're able to make the things you need without making things that you already have that you don't need. We're not going to talk about ribozymes actually. Here is the information if you're interested in them. You may have heard of them before, but I'm not going to ask you about those. Okay, so now you can tell me, what is a coenzyme? Why is enzyme specificity important? What happens to an enzyme below its optimal temperature and above its optimal temperature? Why is feedback inhibition a type of non-competitive inhibition? Actually be able to tell me, describe competitive, non-competitive inhibition, which binds to an active site, which binds to an allosteric site. And we're just not worried about ribozymes. Okay, I'll see you back for part 3 of chapter 5. We're going to start talking about electrons and oxidation reduction reactions and different types of phosphorylation. See you later. Bye.